يعطيكم العافية. We're gonna go through a fairly easy lecture. There are a few foreign objectives here, so nothing really too hard. We're gonna recover and cover our tracks and fill in gaps about the antigen-presenting cells that we have discussed so far. So we know that macrophages have the three main functions. They can do phagocytosis, they are antigen presenters, and they have some regulatory functions in where they produce some cytokines. So let's cover our tracks here. Phagocytosis, this is the regular thing that the same as that would happen in a neutrophil. It will ingest and kill microbes by a phagolysosome. If you recall, have that the foreign material comes from cells through this phagolysosome. The killing is caused by reactive oxygen species, methyl superoxides, as we know, and reactive nitrogen intermediates such as nitrooxides. And then lysosomal enzymes, proteases, nucleases, and lysozyme. All of this will break apart these microbes. Antigen presentation, this is the regular thing that is done on the MHC2 complex to the CD4 cell, and then we will display the B7 protein, which is the co-stimulator for these CD4 cells. Cytokine production, شو حكينا عنه, we know that it can produce interleukin 1, which is a very potent immune activator. حكينا tumor necrosis factor, and other chemokines مثل interleukin 8. So phagocytosis of these all microbes, it is further enhanced when the foreign particle is attached to an IgG, an immune globulin. So an immune globulin, an antibody, هلا بالجزء هاي أول فكرة نحكي فيها عن antibodies. So the first thing that the antibody does, حكينا إنه it is binding to different antigens, and with the portion of the antibody that is hyper variable, so it can bind different. So it can make different antibodies that can bind different antigens, and then there is a constant part of an antibody. The constant part is called the FC portion, the constant portion. يعني فكرها إنه C هي عبارة عن constant. So this is the constant portion of the IgG. This is constant because all phagocytosis that is will bind to this constant portion. So all of them will do the same function, so it would require no change. There's no variability in this change because all of them will have receptors for this portion. When the antibodies cover and engulf, cover and coat a foreign particle, it becomes the charge is different, and then the macrophages will come and then will bind to this FC portion and engulf the foreign microbe. So macrophages have receptor for also. This is the adaptive part. The innate part, the complement, is C3b. It binds the same fashion. This one is not variable to different antigens, but has pattern recognition. So complement will bind to the different patterns that are موجودة بالforeigners. Will bind them and then will be engulfed and taken up by macrophages. So this is an innate part of the humoral immunity. So okay, the internalized foreign particle then fuses within the macrophages with the lysosome. هنا بدخل reactive oxygen species and reactive nitrogen compounds, digest this microbe, and then this fusion with the lysosome with the compounds are two areas where microbes can form defenses and escape being killed. شو يعني? If I prevent the lysosome and the phagolysosome and الفاجوسوم يرتبطوا مع بعض, then it can survive within the phagosome, and lots of bacteria change the cytoskeletal structure تبعت macrophage, so it can stop working on this cytoskeleton توقف الفاجوسوم. It doesn't bind to the lysosome, and this is where a microbe can evade being destroyed and becomes an intracellular, and then it lives inside the macrophage. هلا بالنسبة للانتيجين presentation, will take up most of the time of the foreign particle ingested, degraded, حكينا وتذكروا. So the most appropriate specific parts will remain from digestion, and this becomes the antigen that is going to be presented. So for interaction with the appropriate specific T cell receptor على CD4 positive cell. So how does the cell know, حكينا, which part of the foreign protein to associate? So the cell will use degenerative degradative enzymes, and the less complex parts will be degraded بسرعة. مثلا كانوا so we have a complex sugar and a simple sugar. Simple sugar will be digested quicker. It's the same thing for a polypeptide or a foreign protein. So things that are more complex and وفيهم secondary tertiary structures بتكون أصعب يكسرهم. أشياء فيهم simple structures. هذول simple structures رح يكونوا موجودين بكل مكان. They're not unique, so they will be digested quicker. So this way you come up with a unique part, undigested part of the protein that becomes the antigen that you want to associate with the MHC2 complex. So the complex is then transported to the cell surface for presentation. Cytokine, حكينا interleukin 1, interleukin 8, and 2 necrosis factor, which are important mediators of inflammation, as we said, to prepare the immune system, and then the liver to release proteins. So these three functions are greatly enhanced when macrophages are activated. So if you activate a macrophage, it becomes even heightened antigen presenter, heightened phagocyte, and a heightened chemokine producer. So they are initially activated by substances اللي هم حكينا toll-like receptors, with their toll-like receptor, things that are foreign that we know from patterns. So lipopolysaccharide, مثلا endotoxin. This is the first way where you will engulf and come up. You are coming with in contact with a foreigner, and this is the primary activation of a macrophage. مثلا human DNA is methylated, bacterial DNA is unmethylated. They use something different other than a methyl group. So I would know this DNA, مثلا عندي خلية مكسرة, this DNA comes from a foreigner, so I need to be activated. هذا primary activation. And a secondary activation, this is by gamma interferon that is being produced by مين؟ الhelper T cells. Once antigen has been presented, so they further activate macrophages to do further work. So gamma interferon increases the intercellular mediators that enhance its three functions, which is more antigen presentation, more macrobiocidal activity. And then macrophages migrate and is being promoted by protein part of the complement cascade اسمه C5a. C5a اتذكروا بالA anaphylaxis, so it increases the chemotaxis, بتزيد الدخول. نحكي عنها بمحاضرة الcomplement. So dendritic cells is the second one, alongside B cells that are dedicated professional APCs. Dendritic cells are unique in their function, and they are the main promoters of the primary antibody response. So هذول أكثر تخصصهم أكثر على the antibody response. So the name dendritic points to the cell shape of these cells. They have many long processes that try to بدخلوا بكل يعني المداخل بين الخلايا, and it keeps putting these prongs everywhere, trying to cover as much area as possible. So then the dendritic cells are primarily located under the skin and the mucosa. Langerhans cells من skin اسمهم. And then they stand guard, بسكروا هالbarriers, and then they keep looking for antigens that they can come in contact with. Once they do, then they migrate to the nearest lymph node. تتذكروا the chemotaxis with the CCR7 and the CCR7 receptor. So summary of the interaction here. So for APCs, مثلا dendritic cells, it will engulf and digest the bacterium, CCR7 ذكروا, and then it will find to the nearest lymph node, and then find the correctly specific T cell receptor on the CD4 cell. هلا خلال هذا اللي بصير اللي احنا بنعرفه, at the same time, actual broken fragments of this microbe itself will also migrate. يعني الانتيجين لحاله تكسر ما راح يضل قاعد بالمنطقة. So movement of fluids, movement of lymph, movement of blood, movement of whatever, it will carry these particles. So these particles might themselves be circulated to the nearest lymph node as well. So because of how يعني مش body is not static, the fluids are moving. So if nobody engulfed this broken fragment, this broken DNA, this broken protein, this broken whatever, it will by itself find itself to the nearest lymph node. بالlymph node شو عندي أنا? I have lots of B cells sitting there, and there is a very high chance that one of them will be able to bind to this antigen, and then the antigen will be processed and then presented to the T cell in a T cell dependent pathway. So this is another thing. So we have an antigen presentation of humoral and another one of cellular responses, and then there's a balance between the two. This is not to be confused with follicular dendritic cells. FDCs, although they look similar to dendritic cells, they have different function and location. They are located in B cell containing germinal centers بالfollicles تبعين السبلين والlymph nodes. They do not function as APCs, معناته ما عندهم MHC2 complex, and their job is to capture antigen-antibody complexes. فلما يصير في عندي أنا كتير مثلا an antigen and it's circulating and I made a lot of antibodies, so these complexes بتجمعوا and then they can precipitate, بترسبوا بالأنسجة, and then if that happens, then neutrophils will come and try to engulf and take up these complexes and cause destruction in tissues. So معناته every time you get infected, every time you make antigen antibody, then you will راح تصير ترسب complexes, and then each and every time your neutrophils will start to destroy your body, and that is actually destructive. And this is the mechanism of a lot of destruction, خاصة بالjoints and then certain مثلا connective tissue. So follicular dendritic cells, that's their job, is to capture these complexes. عندهم FC receptor ويمسكوا أي complex antigen antibody, and then they take them up and engulf them. So the antigen antibody complex and then detected by these activated B cells, and the antibody produced by these cells undergoes affinity, ما يسمى affinity maturation. So بياخدوا هدول الcomplexes ما بدنا إياهم يترسبوا, but then I use them for something else. What do I use them for? I improve the maturation of B cells, and then improve the affinity تبع الانتيبودي تبع اللي أنا بعمله ضد هذا الcomplex, trying to make a better antibody and a more specific antibody to this antigen. So هذا أول function. ثاني function حكينا أول function إنه هو engulf the complexes. ثاني function to improve the affinity. ثالث function, they also produce chemokines which attract B cells to the follicles in the spleen, and then this improves their ability to detect them. So يعني it's an added function. Natural killer cells, شو بدنا نضيف عليهم هنا؟ So we know that they kill viruses, infected cells اللي هم بينزلوا MHC1 عندهم, tumor cells that do the same. We also know that they produce gamma interferon that further activates macrophages to enhance their phagocytic function. So this is NK cells are about 5 to 10% of your circulating lymphocytes. So features that so far we want to cover, we want to يعني نجمع كل الأفكار تبعنا. They are large granular lymphocytes. They don't have a T cell receptor. They don't have, obviously that means they don't have CD3 ولا عندهم surface IgM ولا IgG زي الB cell. They do not require being mature in the thymus. That's why they are naturally able to do their job. A normal number in patients who have SCID. So if you recall, that's NK cells is not the affected cell in this manner. So they still have the ability to kill virus infected cells. Activity is not enhanced by prior exposure. So they are part of the innate, more of the innate system. Function of NK cells, حكينا, killing virus infected cells, cancer cells. Killing is non-specific and dependent on foreign antigen, and is not dependent on foreign antigen being presented on class MHC1 or 2. They produce gamma interferon as well, which activates macrophages to kill ingested bacteria. So that will recruit macrophages to these cells that are destroyed إذا في خلية بكتيريا متخبية جوا الفاجوسوم. So this gamma interferon will then activate the macrophage to come up and clean these dead cells that have been infected by this bacteria or microbe. So mechanisms by which NK cells kill the cells that they specialize in policing. So they secrete cytotoxins زي اللي حكينا نعرفهم, perforins, granzymes, much like a CD8 cell. Apoptosis by fast ligand, again the same. The natural part is that without prior exposure, they are able to naturally do their job. They are able to kill without antibodies. However, IgG antibodies improve and enhance their ability, and this is part of the antibody-dependent cellular cytotoxicity mentioned previously in more detail. So this is added, we add this, that we know that antibody-dependent cellular cytotoxicity also affects the job of NK cells to improve their killing and binding ability. هلا بالنسبة لchemokines that activate NK cells, interleukin 12 which is produced by macrophages and interferon alpha and interferon beta. حكينا تذكروا interferon حكينا these improve killing of viruses. So إذا عندك interferons عادة your ability to kill viruses is enhanced. How does that happen? So this is by that by improving my natural killer cells to kill virus infected cells. So interferon alpha and beta are well known activators of NK cells, and so they have no immunologic memory and no T cell receptor. They are independent of MHC protein, tamam. And then this is, we know, down regulation of MHC1 and reduced detection by other cells. So let's see, مثلا, if a cell is expressing an adequate number of MHC1, in this case, if you don't have NK cells, lacking NK cells, predisposes to severe and potentially fatal infection. So if a cell is expressing adequate number of these on its surface of MHC1, they would not kill the cell. So this way you would need a CD8 cell to do the job. مثلا CMV or varicella. NK cells detect the presence of cancer cells by recognizing a protein called mica, which is found on the surface of many cancer cells, but not on normal cells. And then they can actually trigger the production of cytokines and the death of this tumor cell. Neutrophil are very important. Not having them will predispose someone to have a severe bacterial infection. So lacking sufficient neutrophils who have neutropenia بيعمل lots of granulomatous disease. They contain granules in the cytoplasm which give them this staining بتعرفوه من هذا من pathology, I think. And then they have receptors for IgG, and only IgG will facilitate their ability to engulf bacteria. So you know this from your hematology. So as we mentioned many times, they do not have MHC2. They are not antigen presenters, but they can be thought of as a two-edged sword. So the positive edge is that it has a very powerful microbiocidal activity. It can destroy bacteria, and the negative is the tissue damage. نتذكر حكينا مثلا antigen antibody complexes. Any presence of bacteria, they will come and they actually destroy the tissue of this by releasing their enzymes as they are destroying the foreigners. So it's a two-edged sword. That's why مثلا with TH17 I am controlling neutrophils very tightly in the gut so as not to cause destruction of the normal cells, مثلا enterocytes. An excellent example of the latter is the damage مثلا glomeruli in acute post-strep glomerulonephritis. This is the antigen, the antibody complexes. So the damage is caused by the enzyme released by the neutrophils of the glomeruli by C5a activation of these complexes that are deposited in the glomerular membrane. Eosinophils, تذكروا حكينا عنهم, parasites and antibody-dependent cellular cytotoxicity. So eosinophils, they have these red colored or pink colored granules. So parasitic infections, you need them, and hypersensitivity diseases, this is where eosinophils are implicated. So what do we know of their function? They can bind Fc portion of IgE and IgG and defend against مثلا larvae or parasites, and they can release their content on these parasites and kill them. And they can contain histamine, which is يعني هم هذا has the effect to aid in the control of immediate hypersensitivity reaction تبع الهستامين. So they contain histaminases عفوا that breaks histamine اللي هو موجود بال IgE dependent release of histamine from histamine مثلا releasing cells مثلا mast cells or others. تمام. The granules also contain leukotrienes, peroxidases, which can damage tissues and cause inflammation as well. And these also granules can contain major basic protein which damages respiratory epithelium. بتذكروا بتعرفوا انتم كمان انه the presence of eosinophils and high eosinophils is part of the pathogenesis of asthma. So this is due to the production of major basic protein, which is the protein implicated in the damage of the epithelium in the respiratory tract. They can phagocytose bacteria, but obviously they're not antigen presenters. Actually, phagocytosis is يعني صارت becomes a spectrum. So lots of cells are able to engulf foreign objects if they have the ability to destroy them. So يعني they're not really, but they have this ability. And you know this from your hematology, how eosinophils look like. Basophils and mast cells. So these have the basic dye بيصير بيطلعوا positively charged بيصير لونهم أزرق. So again, due to the granules that they appear blue, they circulate in the bloodstream. Mast cells زي بيشبهوا basophils, but they are fixed in the tissues, and both have receptors on the cell surface for the FC portion تذكروا لل IgE, and this is where the release of histamine happens لما تصير في cross linking of IgE. So we know this. So they have receptors for the FC portion of the heavy chain of IgE. Lots of IgE can cross-link and then release their mediators, histamine, which is a potent anaphylatoxin, and then enzymes مثل peroxidase, hydrolases, and others are released. They cause inflammation, and then large amounts causes severe immediate hypersensitivity. This is type 1 hypersensitivity and causes anaphylaxis. Mast cells also play a role in innate response to bacteria and viruses. How they have toll-like receptors that recognize bacteria and viruses, and this anaphylaxis can improve. يعني in a small amount, it improves the entry of blood and migration of cells, so they can increase the detection of these foreign microbes. So they can respond by releasing cytokines and enzymes, and then بيعملوا inflammation, and they attract neutrophils and dendritic cells and macrophages to come and then clean up the area. That's the principle of histamine, really. But if you have too much of histamine, becomes systemic anaphylaxis. Dendritic cells are important. Is that they can initiate the adaptive response. So the role of mast cells in inflammation has been demonstrated مثلا بالrheumatoid arthritis. And they can produce both inflammatory cytokines and enzymes that degrade cartilage in joints. So they are implicated here. Eosinophils implicated بالasthma. Basophils and dendritic cells are mast cells are implicated بالrheumatoid arthritis. So from we know now, pro-inflammatories, we know this is just a slide. مثلا interleukin 1, 6, and 2 molecules, and 10 and transforming growth factor are anti-inflammatory cytokines. So if you look at the slide, هلا صرنا نجمع احنا افكارنا نجمع the cytokines that we know so far. So from macrophages, so this is what we have so far. I'm not going to read this. I'm sure you can. We have mentioned everything here. So this is a very good table to keep track of everything. So from pro-inflammatory cytokines of the macrophages and what they do. So anything that we didn't, everything here so far we mentioned in other areas of our lectures. So cytokines affecting lymphocytes. Okay, so interleukin 1 and 2. So the remainder of the lecture is just goes through these 3 slides is just the interleukins that we have so far. So interleukin 1 حكينا potent activator. It's an endogenous pyrogen. Interleukin 2 نعرفوا إنه this is the activator of helper cells and CD8 cells. نعرف interleukin 4 and 5 for the Th2 response. Interleukin 5 also induces class switching to IgA. Interleukin 6 is a pro-inflammatory cytokine. حكينا this is production of acute phase proteins. Interleukin 7 بجوز ما حكينا عنه, but it produces stromal cells in the thymus. It's maintained to push stem cells to keep differentiating. I think we mentioned this in the T cell receptor, in the T cell maturation. Interleukin 10 and 12, they regulate. حكينا this is anti and this is pro. This regulate production of Th1 as well. And interleukin 12 and gamma interferon X is very important to the ability of our host defenses to control true intercellular pathogens. So, مثلا TB, listeria, and others. Okay, so interleukin 13, we haven't mentioned this. This is implicated in so interleukin 13 would be this is shown to be necessary to cause asthma. بالنسبة ل transfer growth factor beta, we know this is an inhibitory one, and it then regulates the immune response. So it wants to improve healing and synthesis of collagen, and to do this I would want to down-regulate my immune response in a certain area. So this is the growth area. So بخلصت inflammation, I want to go to growth. I will go to transforming growth factor beta. So cytokines that affect macrophages and monocytes. So chemokines, these are chemotactic cytokines that function attracting phagocytes. They are produced by various cells, and such as endothelial cells. And because ليش endothelial cells? Because these are the ones that are going to capture the macrophages to send them into the area of inflammation. هذا من محاضرات الباثولوجي بتعرفوها. So this attracts circulating neurophages and macrophages to the site. We know that the interaction between the ICAM protein facilitates the movement of white cells into the tissue, so they can migrate into the tissue. So the alpha chemokines, مثلا interleukin 8 is an important alpha chemokine produced by mononuclear cells, which attracts other neutrophils that are produced by activated مثلا lymphocytes or monocytes. The beta chemokines are produced by activated T cells. They attract macrophages and monocytes. RANTES and MCAF are important beta chemokines. How do they work? مثلا neutrophils, monocytes, they have receptors for these molecules, and إذا الكيموكاين موجود, it will interact with the receptor and cause cell surface protein to change. Now هدول الخلايا بصيروا now they're able to adhere and migrate to the endothelium, to the infection site. Cytokines affecting PMNCs, مثلا leukocytes, tumor necrosis factor activates the phagocytic killing and activities, we know this, and increases the synthesis of adhesion molecules to improve adhesion to endothelial cells. Mediate attachment طبعا. Chemotactic factors مثلا neutrophils, basophil, eosinophil, or are selectively attract each cell type حسب الانفكشن اللي عندي. Leukocyte inhibitory factor inhibits migration and then to turn off this process. نعرف interleukin 17 how it recruits neutrophils and controls them in a site of infection in the gut for mucosa. Interleukin 3, this is produced by T cells, helper T cells, that improves the proliferation of growth of stem cells in the bone marrow. So you want as much as much as you are meeting and using helper T cells, as much as you are going to be proliferating and growing your stem cell to make more and more cells. فمعناته in a perfect world, أو مش perfect world, but if you control infection at all, خلايا كل الخلايا proliferation of stem cells, white blood cells بصيروا يقلوا, because as you are not activating helper T cells, you are reducing interleukin 3 and then you're reducing. فبتلاقي البني آدم اللي بيلاقي infections أكثر بحياته عنده more interleukin 3 and making more stem cells into bone marrow cells into white blood cells مثلا. Okay. Cytokines that are produced by macrophages that affect other cells. We know tumor necrosis alpha is a pro-inflammatory site مثلا. هذا بيختلف بالجهتين موجودة بالتيبل هاي at low concentration increases endothelial cell adhesion. أنا بتزيد الادهيجن. I'm activating the respiratory burst تبعت النيوتروفيل بيعرفوها هاي and the oxygen species, and it induces fever. So it is a pro-inflammatory. At high concentration, it is an important mediator لإشو لإندوtoxin induced septic shock. So high levels of TNF is what it is عفوا implicated in septic shock. تعرفوا مثلا systemic vascular resistance in all types of shock is vastly increased. So مثلا hypovolemic shock, you are increasing your systemic vascular resistance, مثلا cardiogenic shock or whatever. In distributive shock اللي هي نوع منها اللي هو septic shock, we see that this is the only one where systemic vascular resistance is actually reduced. وهذا بيساعدنا مع الفيفر هنا انه نعرف انه fever, and this tells you that this is مع shock مع low blood pressure, this all will point to you towards a distributive septic shock مع vasodilation تبع nitric oxide. So this is an important way this happens. How? By tumor necrosis factor alpha and high concentration. Other effects of cytokines: interferons, they are block virus replication, alpha from leukocytes, beta from fibroblasts. They are induced in by viruses, and they exert a very powerful antiviral activity by يعملوا ribonucleases that degrades viral mRNA. معناته inhibit replication of translation of viral proteins. They also activate NK cells, which حكينا هذا, this is needed to improve their ability and to look for in certain area to look for these cells. Gamma interferon produced by Th1 أكيد, and is the signature cytokine involved in the inflammation mediated by these cells. هذا هو الجاما interferon is a very potent activator of the phagocytic activity of macrophages, NK cells, and neutrophils. كل هذا to improve their ability to do their job. يعطيكم العافية. We will stop here, and again we will meet for any questions whenever you guys want to do so. يعطيكم العافية.